Alright guys, welcome to Subu Medicine, and this is male reproductive systems. So today we'll be discussing about the physiology aspect of the male reproductive systems, okay? So whenever we discuss about the male reproductive systems, we always have to talk about, what is this? This is going to be my hypothalamus, right? You guys know this by now, okay? Hypothalamus, right? So whenever we discuss about this, we have to talk about the hypothalamus, and what is this? Look, this is hypothalamus, this is anterior and posterior, right guys? You guys know this by now. So this is going to be like, okay, your anterior pituitary, right, this part. And this is going to be posterior pituitary. So we always have to talk about this hypothalamus anterior. And then this drawing right here, you see this structure right here, guys? Like this part, like from all the way down, look, from all the way down, all this stuff, okay? You can imagine this is as a testes, okay? Because we're going to discuss about the spermatogenesis, okay? And whenever we talk about that, we need to talk about the testes, okay? Because testes is the one that is important for synthesizing your sperm productions for male, okay? So these are different cells that we'll talk about it. But we have to discuss about the hypothalamus anterior testes axis, okay? Now, what is going to happen here is that, okay, so see this nucleus right here, this nucleus is called arcuate nucleus, okay? It's called arcuate nucleus. And, this arcuate nucleus has a cell bodies to synthesize, the hormone called gonadotropin releasing hormone, right? So, there are other nuclei too, like for instance, let me write down here. Ventromedial, the ventromedial or dorsolateral, okay, all of them also have cell bodies to synthesize this gonadotropin releasing hormone. But remember this, just remember this arcuate nucleus is very important, okay? So that is taking place in a hypothalamus, right? And they actually synthesize this protein called, let me just write down, okay? It's a ligand that gets synthesized here, which is called kisspeptin. Because this kisspeptin, all right, this is the one. The kisspeptin is the one that this is a ligand, all right? This comes and binds here with its receptor right here, in the anterior pituitary, okay? And here they have a receptor for this, okay, which you can also say gonadotropin releasing hormone for our case. They have a receptor on these cells, and these cells are located in anterior pituitary. What are these named cells? So you see the blue, these are blue cells because they take basophilic stain. Okay, we call them a gonadotropes, okay? So these are gonadotropes, all right? And they're basophilic in nature. So whenever these guys come down here in the cells, okay, they are associated with ZQ pathway here, okay? So let me just write down. These gonadotrope cells, okay, if I make a cells right here, okay, and then this is a cells right here. So when it comes, let's say gonadotropin releasing hormone, okay? So this is come down here, binds with these cells, okay? And these cells are associated with here with the ZQ pathway. And you guys know what ZQ leads into, okay? This is going to ultimately, okay, let's just write it down here. Let's make these cells a little bigger. So you guys know this, what I'm talking about here. All right, let's make these cells bigger. So basically right here, okay? Let's make these cells bigger. Right here, okay? Here what is going to happen, the ZQ pathway. Okay, this is going to ultimately what? Activate, okay? I'm not going to go super detailed because you guys should be knowing for now. It's called IP3, and then other one is DAG, right? And then DAG activates what? Protein kinase C, right? That's what it does. And remember, these guys go to, let's make this a nucleus right here, okay? There's a nucleus right here. And then they're going to stimulate a gene, okay? And then, synthesize, a very, very important process, because this is going to take place here. And I'm writing it down. One is called FSH, okay? And the other one is called LH, okay? FSH stands for what? It's called follicle, okay? It's called follicle stimulating hormone, okay? And this is called luteinizing hormone, okay? So, so basically these cells, okay, this gonadotrope cells. Let's write down the gonado, okay? It's called gonadotropes. These gonadotropes are important for synthesizing FSH and LH, okay? Follicle stimulating hormones and luteinizing hormones. Because these guys are going to regulate the testes, okay? And how it's going to regulate testes, that's what we're going to discuss about it, okay? So basically, I'm just talking about, we always have to talk about hypothalamus. What is this? So let me write it down here. It's a hypothalamus, okay? It's going to be a hypothalamus, right? And there's anterior, okay, testes axis. Because we're talking about the male, so we're talking about testes. If it's a female, we'll say ovary. Or in simply, sometimes they say hypothalamus anterior gonadal axis, all right? Now after this, we have to go a little more depth about this, okay? So now, hold this thought here, all right, guys? So, come here and we'll talk about this testes first, all right? So, you guys know by this now, right? The testes, what do testes have? The testes has different cells there, right? So, you look at this. These are blood vessels right here, all right? These are blood vessels. And then, this part right here, this part, okay? Let me make this. All this part right here, all right? This is going to be your interstitial, okay? And these are going to be cells after that, okay? These interstitial, okay, have cells there. They're important cells, okay? These cells are very important cells. Let me write it down here, okay? These cells, you can simply call these pink cells, okay? You can call this a lytic cell, okay? Or you can say these are interstitial cells, whatever you like to call, okay? These pink cells are called lytic cells, and they're located in where? In the interstitium. That's why we call them interstitial cells or lytic cell, whatever you would like to call. And you see the green cells right here? There's also these green cells are present here, okay? And you have to remember this, And these cells are called myoid cells. Let me write it down. What should I? Okay, let me put it down here. Look, these cells are called myoid cells. They're also, okay, these are called myoid cells. And the functions of the myoid cell is that these are smooth muscle cells. And what they do is that, they do contractions here. Okay, you know why they do a contraction? Because they're smooth muscle cells. So they contract, okay, to secrete out your sperm. All right? And we'll talk about how they do it, okay, later. And we'll also talk about what is the functions of the lytic cell too, okay? Now, but remember, this is a testis, but this is where is happening here is that this is called seminiferous TV, okay? Because remember, seminiferous TV is the functional unit of the testis, okay? So if I quickly, let me write down here, okay? If I make this like a testis right here, okay? All right, this is a testis. What is going to happen here is that, okay, there's going to be cell, there's a convoluted part. This is called your seminiferous TV, okay? And then the seminiferous TV, they have this lumen right here, right? On here, right here, okay, right here, these are the one, okay, these pink cells are outside of this seminiferous tubule, and these pink cells are lytic cells, and they also have a myoid cell, okay? So you have to remember this. But remember, I just drew this stem cell, this seminiferous tubules and draw here, okay? And then this is, let's say this is a seminiferous tubule, all of them, and there's a lumen right here, okay? There's a lumen, and then outside of the seminiferous tubule, what do you see? This interstitial or your lytic cells, and also you see myoid cells, okay? That's what it is. But in the seminiferous tubule cell, this is a seminiferous, okay? You can think about this, the white part is, you can say this is a seminiferous tubule, okay? And there, you're going to see these cells. Like, what are these cells? These are very, very important cells, okay? These cells, okay, these are called, the different names are given for these cells, okay? I'll write down here. Okay, let me write down here. These are called Sertoli cells, all right? Or they also called sustentacular cells, okay? Sustentacular cells. These are different nomenclature for these cells, okay? Sertoli cells and sustentacular cells. Or they also, because what they do is, their function we'll discuss, because they are providing nourishment to the the sperm cells, okay, to those germ cells to synthesize sperm, okay? So because they provide nourishment, we also call them like a nursing cells, or you can say nurse cells. Let me write it down. Let's say nursing cells, okay? So you can simply call all of them, okay? Because remember, the Sertoli cells are very, very important functions here, all right? Now, then after that, okay, not only the Sertoli cells provides the nourishment to this sperm cell, okay, or those primordial germ cells, the cells that has to synthesize, your sperm productions, or another name for sperm production is spermatogenesis, okay. What the Sertoli cells do is, let me write it down, couple. They produce, okay, obviously they are going to, okay, and obviously this is happening on the influence of a FSH, and we'll talk about this, okay. They are going to, okay, secrete very, very important some of the hormones. Like for example, one is called androgen binding proteins, okay, and we'll talk about what they do. Not only that, they also secrete something called inhibin B, okay, and MIF, Mullerian inhibiting factors. Not only that, they're also very important for secreting growth factors. Okay, and then also like other stuff like cytoplasm or ceruloplasmin, okay, that is for the copper transfer. Even for the iron, like transferrin, they also synthesize all of those, all right? Now, hold that thought here right now, okay? These are some of these, functions of the Sertoli cells do. But remember, one more the Sertoli cells do. You see this, what is this? These are bridges here. You see this? These are called cytoplasmic bridges, okay? And these two cells are called the Sertoli cells. These are Sertoli cells, or sustentacular cells, meaning what? Supporting cells, okay? And they're providing these bridges, okay? And these bridges are what? Formed by adjacent cells, okay, of the Sertoli cells. They're making the Sertoli complex, okay? And what that does is that makes this blood-testis barrier, okay? That's what it does. Blood-testis barrier, that's what it makes, okay? Because remember, your blood cannot have a direct connect with the cells that has to do with the sperm production. If that does, then what is going to happen is an immune reaction will occur, okay? And that will destroy the sperm production, all right? So that's the reason why, it is so beautiful that this Sertoli cells creates this something called blood. Let me write down here. Okay, let me write down here. What is this called? It creates something, it makes something called blood, okay, testis barrier, all right? Very, very important concept, guys, here, all right? So these cytoplasmic bridges are called, okay, that has been, established by the sustentacular Sertoli, or this nursing cells, okay, that is called blood-testis barrier. Beside that, it is also providing the nourishment, okay, and also it is also synthesizing these different types of hormone, okay, because all this play a very, very, very important role, and we'll talk about that. Now, all right, after this, we have to go a little more depth about this, all right. Let's talk about this thing. Let's say there's a germ cells, okay? Let's make here germ cells. We have to make a germ cell right here, okay? And remember, these are primordial germ cells, which means that this gets here, okay? What is happening? These primordial germ cells, okay, around, okay, in the early development, okay, what is going to happen is that these primordial germ cells, they migrate from the yolk sac from your mother, and then they go to testes and sit there, okay? Remember, this primordial germ cell does not get differentiated, okay, until you hit the puberty, okay? So that's something that you have to remember. Oftentimes they ask you that questions, okay? It's a primordial germ cell, okay, which does not get differentiated until, you hit the puberty, all right? Now, these primordial germ cells, okay, when it gets like here, let's say in this area, what is this area called? In testes, okay, there's a name is given for this, okay? We call these cells, spermatogonia, all right? Let me write it down this. What do we call this? It's called Spermatogonia. And this is a plural form, okay? If you want to call it singular, you can say spermatogonium, all right? Now, the cells is called spermatogonia, you know. And when you hit the puberty, okay, what is going to happen is this. These cells gonna grow, okay? The mitosis will occur, all right? So let's just make these cells, all right? And then I made a nucleus right here, okay? And then in the nucleus you have, oh, okay, what is this? On top of this, look, you have this DNA right here, okay? There's a DNA right there. You see this? All right? What is gonna happen is mitosis will occur. Let's write it down here, okay? The mitosis, I'm gonna make this here like this, okay? Mitosis means growth will occur, okay? And then remember these cells, okay, this primordial, this spermatogonia, these are stem cells, okay? You have to remember this. These are stem cells. So because they're stem cells, and you guys know the property of the stem cell, what are they? You know the stem cell, what they do? They have two functions right here, okay? One these guys do is, okay, let me write it down here. One they do is like, you know, they will maintain their own property, okay? And at the same time, okay, they also give rise to different other cells. You know, that's the property of stem cells, right? So, maintaining their own population, okay, let's just make this. They maintain, okay, so they maintain right here its own property, okay, let's just make it similar to like that. And then we'll use this, like this. And then they also make their clone out of it, okay? And then clone out of it. Look at this, okay? So this process happens, okay, because that's the proper stem cell. It does a symmetrical and a symmetrical way, okay? What does that mean? Meaning that it makes a clone out of its own cell and also give rise to different other cells. That's what it does, okay? So when this does that, okay, this is a mitosis, or this is a growth process. What is this process called? We call it a growth process, okay? This is a growth process. And when that happens, okay, this gets differentiated into two types of cells. These cells right here, okay, which is making the clone out of cells, we call them a type A cells, okay? And these cells, okay, which is differentiated into daughter cells, we call them a type B cells, okay? Or there's another name given for this type A cell, we call them a dusty cells, okay? Remember guys, you always get extra here, right? Because that's the beauty of this Subu medicine. You know, you always get extra here. And what is this type B cell? It's called crusty cells, okay? And the reason why they call this crusty cells and dusty cells is based on the chromatin pattern that is present in this nucleus. If you see a type A cells, okay, the chromatins there, they're gonna appear to be more like a pale stained chromatin fibers. But if you see more like this type B, the crusty cells, okay, the chromatin patterns are gonna be those, you know, the coarse granulated, heavy chromatin fibers that you're gonna see in the type B or the crusty cells, okay? So these names are given based on the crusty and dusty. They're given on the patterns of the chromatins, all right? Now, so now what we're going to do is we're going to focus on the type B cell. Remember, this type A cell it's maintaining its own populations, okay? But this type B cell differentiates into different cells, okay? So that's what we're going to focus on. Remember, this type B cells, this is happening in the basal area. Look, this, you can think of basal area right here. This is more apical area, right? Now, what is going to happen is this type B cell is that, you know, this type B cell can secrete some of the proteolytic enzymes. So they secrete proteolytic enzymes. And this proteolytic enzyme, what is going to, they're going to this, you know, break these bridges, okay, these cytoplasmic bridges that has been formed by these Sertoli cells, all right? So now, what is going to happen? This type B cells, well, across the cells will come down here, okay? So remember, when this happens, okay, we no longer call them a type B cells. We call this part, okay? We call this part, let's say, here, right here. We'll make these cells, okay, right here. And then I'm going to just make a DNA here, okay? Like, because remember, it grew, so it's going to have we'll make it, okay? It multiply like this, okay? So we'll make like a bunch of them, like that. Okay? Now, these, we call them a, okay, let's write down. We call them a primary, okay, we call them a, it's called primary, okay, spermocyte. Look, this is a primary spermatocyte. Look, if you look at this, it's spermatogonia to getting the primary spermocytes, okay? This process is called mitosis, okay? The growth is happening here, okay? Replication is occurring here, all right? So, here, if you see this, okay, in here there's spermatogonia, you know? Like, let's write down here. What is usually happening there is that, okay, usually it has a what? It has a 1 chromosomes and 2 chromosomes. Look, it is going to be look like that, right? So basically, you got a 1 chromosome from a father, you got a 1 chromosome from a mother, right? So basically, you have what? Diploid number of chromosomes, right? That's what it is. So you have a diploid number of chromosomes, which basically means is that you have a 2n. What does that mean? 2 basically means it's a number of chromosomes, right? So you have a 2, and there are 23 of them. So we call them there's a 46, okay? There are 46 chromosomes there, and that is, we call them a diploid, okay? And remember, because these chromosomes, okay, these chromosomes, or we determine number of chromosomes based on the centromere, okay? And centromere is, let's say, this is the middle portion right here, okay? We can say this is the middle portion, which is connecting to, like, you know, connecting this upper and lower part, all right? Now, these number of chromosomes, right? Based on the number of, like, centromere, we determine the number of chromosomes, right? But remember, we also have to talk about what's the DNA here, okay? Because DNA is usually, okay, we usually use big N, okay, which basically tells the notations. These are notations of what's the DNA that's present in each chromosomes, okay? Because there are 2, because there's a 1 here, 1 here. So what is, that's also going to be 2. This is going to be look like this, okay? So you have a 2N. This small n is the number of chromosomes, and this is a 2n is a notations for the DNA that's present in here. For this is spermatogonia, okay? But now if you look at this, what is happening here? The primary spermatocyte, it's still, okay, I'm gonna go this upward. Here, look, it multiplied, right? It duplicated. So, but it's still, look at the centromere. There's still 1 here, 1 here, 2, right? So because only 2, it's still a 2n, okay, still 46 chromosomes. But remember, it multiplied, right? So because it multiplied, this DNA replicated, the DNA also multiplied, okay? So because of that, DNA is level what? We'll write down, it is going to be 4n, okay? So this is a primary, okay, let's just write, primary spermatocytes, all right? Now, after this, let's just talk about. Now, then after that, look, let's make this arrow, let's go upward like this, okay? Here, what is that going to happen is that, now after this, okay, the process that will occur is called meiosis, right? Remember, I'm not going to go super detailed about this, okay? It's called meiosis 1, but this is the time where, okay, what is going to happen here is that, you know, the prophase, anaphase, or the metaphase, the telophase, those are going to take place, okay? And then, like, cytodifferentiation will occur, okay? And resulting into, okay, let's make this, resulting into, let's make a 2-part right here, okay? 1 here, 1 here, okay? Because this is going to reduce right here, and we'll just make it, all right? Because they do all the crossovers and all that can happen, we'll just make it. I'll just write it down here. What they'll make here is that 1 here, 1 here. 1 here, 1 here, like that. Okay? So, hold this side, because in this one, there is a mix of both of this 1 chromosome. There's a mix of both paternal and maternal, okay? Because there's a lot of this crossing over that can happen, okay? So, because of this, okay, because they reduce from, because this is called, meiosis usually is a reducing, okay? You can think about reducing. So, in this process, what is the primary spermatocyte? This is reduced into haploid, all right? It's called haploid. So haploid, and it became, okay, 1 small n, all right? That's what it's become, 1n, because it's not the upper lower, so it's going to be small, 1n, because it's going to be a 23 number of chromosomes there, okay? But the DNA also is split in half, right? So because of that, look, we say this as, let's say, we can simply say this is going to be look, we can simply say this is going to be a 2 with the big N. Okay, so this is a 1. Okay, let me write down here. 1 small N right here, and then 2 big N. Okay, and this big N is representing the notations of the DNA that is present here, that is present in this. Okay, and this is called secondary. All right, it's called secondary spermatocyte. Okay, this is called secondary. From the primary, you go and make secondary spermatocytes to the meiosis process. Okay, now what is going to happen ultimately is that now let's make this, okay, from both of them. All right, the last two, again, this part we call them a meiosis. Okay, it's a meiosis. Okay, 2. These secondary spermatocytes will further go, okay, remodeling or modifications, and ultimate result is, let's just write down here, okay, 1, 2, 3, and then 4 here. It will basically, okay, here 1 here, we'll make a 1 here, and we'll make a 1 here, and we'll make a 1 here. Okay, look at this. This will result into 1, 1, 1. So you have a 1, it's a haploid again, and the DNA is what? 1 big N. Okay, this is okay, with this meiosis 2, okay, the further remodeling happened, and these secondary spermatocytes to this path, these are called now, this is called, let me write down in a different color. Okay, what should I write down? Let me write it in red. This is called, okay, you can say this is a spermatids. Okay, that's what it is. Now, even from the spermatids, there's a further modifications happens. Okay, further modification happens. Basically, what happens is that, remember, these guys, okay, this is taking these guys, happening in the endoplasmic reticulum right now. Okay, all this, and what is going to happen in the Golgi? This is where they get those, because remember, what are we trying to make? We're making a sperm, right? So those, look, let me write down here, like a acrosome, okay, capping, all that is gonna take place in like a Golgi, okay, acrosomal reaction, also the forming of those, flagella and all, okay. This is gonna happen, okay, and then ultimate result is you're gonna make a, okay, spermatozoa or sperm. So this person, the spermatid, you're making your spermatozoa, which is a mature form, okay, by putting a acrosome cap and putting a flagella. The sperm now, finally, it will look something like this. Now if I make it like this, and remember, this is head, so this is right here. There's also vacuole here, and this is going to be my mid piece, right? This is, we say head, we said this is a mid piece, and this is a flagella, this is a tail right here, okay? So this is going to come out like this, okay? So we'll make this. Look, we'll make this bunch of them. Sorry for the drawing, but you guys know what I'm talking about. So this is what it is, okay? So now, this process, okay, these are different phases, we said, okay? So from the process, whenever you make from this your first spermatocytes to spermatids, okay, from all the way down, look, from the spermatids to like, let's say this part right here, okay, what is this? We call them, okay, we call them spermatogenesis, okay, let me write down here. What do we call this all phase? We call them a spermatogenesis, okay? That is from where? From the primary, okay, first primary to spermatids, okay? So we call that process called spermatogenesis, guys, okay? You have to remember that. But from the spermatids to spermatozoa, or the sperm, the mature sperm, okay? Because this is going to get mature right here, right? This process is called okay, let me write it down right here, okay? That is called spermatogenesis, okay? Remember, guys, there's also another name is given for spermatogenesis. You can call this meiosis too, if you like to call, okay? Whatever you like to call. But remember, guys, do not get confused. Spermatogenesis with, okay, let me write down here, okay? This one. Do not get confused. Spermiosis. Do not get confused with this. Because spermatogenesis with the spermiosis is a completely two different things, okay? Spermatogenesis is, as I said, you're just making mature sperm, okay? But, this sperm is matured, when it becomes spermatozoa, what is happening is usually these sperms are embedded with this, like this. What is this? With this Sertoli cells, okay? And the process in which this, this spermatozoa or sperm, okay, that gets secreted out to the lumen, okay, that is called spermiosis, okay? It has to leave this Sertoli cells because right now it's embedded in the apical surface of this Sertoli cells. The process in which these cells leave, the sperm cells, they leave the Sertoli cells and go into the lumen, okay? The seminiferous lumen, that is called spermiosis, okay? Now that you have to remember that. Now, look, all these things, okay, that you guys have to draw and try to understand this, okay? But remember, guys, these guys, okay, the spermatogenesis would not take place if there is no help of, come back here, look, this guy, what is this called? FSH, follicle stimulating hormone. Because this guy works on the Sertoli cells here. They have receptors here for the Sertoli cells here, all right? Look, you can remember like this. There's an S here, right? If the S is there, then you can think about it. It works at the sustentacular cells or the Sertoli cells. And there's L here, right here, right? LH. You can think about this works at the lytic cells, okay? And this works, this FSH here, okay? Let's say the FSH comes here, okay? Into the blood, and it goes to these cells right here, okay? Let's write it down here in the cells. Let's make the cells right here. In this cell, they have a receptor right here, okay? This FSH receptor is associated with the GS pathway, okay? And you guys remember, with the FSH binding the GS pathway, what is going to ultimately result into, okay? Remember, you are going to activate what? Adenyl cyclase, right? And then from there, what is the formations of? ATP into cyclic AMP and then protein kinase A, right? And this protein kinase A, right? This is going to go to the nucleus of the cells, okay? And it starts synthesizing all this guy, enzyme binding proteins, inhibin B, all that kind of stuff, okay? Not only that, these guys will also, create the activity of, stimulate the activity of all this, stimulate the gene, okay, to activate the process of all this process of forming a sperm, okay? So these guys will work into, okay, all this part, okay, all this part, right here, all of them. Just remember that. And that will what? Create your sperm productions, okay? Now, one thing that you have to remember is that, what is this inhibin B? This inhibin B is a, okay, let's write it down. It's a negative feedback mechanism, okay? For what? What it does is that, okay, whenever this inhibin B is producing, meaning the FSH is coming in the Sertoli cells and synthesizing all that, right? This inhibin B, okay, what they do is that it comes here, look, it comes in the hypothalamus, okay? And this inhibin B, you know what they do? Let me write down here. Inhibin, okay? What it does, it comes here. That's what it does. And also in the hypothalamus, okay? It is a negative feedback, okay? So basically what it does is that it, based on the level of inhibin B, okay, that regulates the productions of the FSH Okay, so if it is a high level in heaven, we don't want to make, okay, we don't want more FSH to release here, okay? So what is that? It regulates the anterior pituitary to produce less FSH, because FHR are going to come and work on the Sertoli cells to do all this property, all right? So in heaven B is the negative feedback, all right?